Hello and welcome back to our session dedicated to the immune system. The immune system can be divided into two major parts, and the first type of immunity is the so-called innate immune system. This immune system is also referred to as the non-adaptive immune system. It represents the first and second line of immune response and also relies on mechanisms that exist before the infection is established. In contrast, the acquired or adaptive immune system is the third line of response if the innate immune response fails, and it relies on mechanisms that adapt after the infection is established. In terms of the timeline, the innate immune response is the first type of immunity which is activated after the entry of a microorganism into the system. If a certain threshold level of antigen or microorganism is present in the system, this leads to activation of the innate immune system, which leads to the so-called inductive phase. After inductive phase is completed, the adaptive immune system kicks in within the so-called effector phase. After effector phase is complete, there we have the so-called memory phase, but this is something quite specific for the adaptive immune system we are going to be learning more about next week. So let's go back to the innate immune system. Again, we have three lines of defense. And the non-specific innate immune system includes chemical and physical barriers. This is the first line of defense. In addition to the barriers, the innate immune system also includes systemic responses such as inflammation, which is the second line of defense. So, what are the mechanisms included in the first line of defense? We have the skin, which is the most obvious physical barrier between the organism and the environment. We have also ciliated cells that are present in our respiratory tract, which we have ciliated cells in our nose, but also in lung epithelium. First line of defense also includes molecules, including lysozyme and mechanisms such as coughing and vomiting. Those are all responses that belong to the first line of defense. The second line of defense, also belonging to innate immune system, the formation of blood clots if a pathogen is detected by the system, release of histamine by mast cells, release of cytokines by macrophages, and action of leukocytes. Those are all very specific mechanisms and barriers that belong to the innate immune system. In contrast, the specific adaptive immune system is the third line of defense against invading pathogens. It is mainly mediated by lymphocytes and involves antibodies and T cells. So today we are going to be hearing more about the innate immunity, whilst the adaptive immunity is going to be covered in the videos next week. So, innate immunity. Innate immunity is present from the birth in all individuals and based on a very specific genetic makeup. It relies on already formed components, and it is fairly rapid in contrast to adaptive immune system. So the first response is present within minutes after infection. Innate immunity is also not very specific because the same molecules and cells respond to a range of pathogens. So regardless of the nature of the microbe or microorganism that is infecting the system, the same set of molecules and responses, including inflammation, are usually used. Another important characteristic of the innate immunity is that it has no memory. So the same response is going to occur even in case of a repeated exposure. And this is the case in terms of the time of activation of the innate immunity, but also regarding the strength of the immune response. If innate immune system is fighting a microbe, both the timing and the strength of the response is always gonna be similar. If we now look a bit closer at the mechanism, and more specifically at the first line of defense, we will see that the most obvious physical barrier, the skin, serves as a barrier that prevents the passage of microbes and is also quite acidic. The pH of the skin is between 4.5 and 5.5. Moreover, the surface of the skin contains good bacteria that can outcompete pathogen, and this is quite important. In addition to this, the sweat also contains bacteriostatic substances, for instance, lactic acid. In addition to the skin, the respiratory passage is lined with ciliated cells and covered in a layer of fairly thick mucus. The mucus traps invading microbes and cell debris, and cilia remove those particles. Within the stomach, the acidic pH destroys most of the foodborne pathogens. So this is another first line of defense mechanism, which is quite important. In addition, similar to the situation on the surface of the skin, in our stomach, good gut microbiota outcompete pathogen under physiological circumstances. And this is actually something quite relevant if you think about side effects of antibiotics. If you prescribe antibiotics to a patient, usually not only pathogens are affected, you will always also affect the good microbiota. In addition to barriers and pH and microbiota, also sweat plays an important role in preventing infections because it simply washes away microbes. Also, the slightly acidic pH of the skin slows down bacterial growth. Cerumen, earwax, protects the ear canal by trapping particle, and last but not least, saliva and tears contain an enzyme called lysozyme. Lysozyme ruptures bacterial cell walls because it catalyzes the degradation of the peptidoglycan matrix, and this is something you probably listened about in your microbiology lecture series delivered by Dr. Black. In addition to those barriers, innate immunity also relies on molecules. So this so-called humoral mechanism include lysozymes and the complement system. The complement system is a group of proteins that are found in the blood plasma. In contrast, the complement protein work with antibodies, so they sort of cross-couple the innate and the adaptive immune system. They complement the action of the antibodies. They mediate the lysis of the microbes and mark them for phagocytes, cause inflammation, and also stimulate mast cells to release histamines. If we now look a bit closer at the complement cascade, we will notice that attachment of an antibody to a bacterial cell triggers a response by complement proteins circulating in the blood. So those proteins complement antibodies that already have bound to the microbes. Those proteins interact with the bacterial membrane and form holes in it. Formation of those holes disrupts cellular metabolism of bacteria, and in addition to this, complement proteins may also disrupt membrane integrity to the point where the whole bacterial cell ruptures. However, if the pathogen is able to pass the first line of defense, this leads to infection. In this case, the second line of defense kicks in and deals with the pathogen. The second line of defense is cellular and involves white blood cells, including natural killer cells, neutrophils, macrophages, mast cells, basophils, and also eosinophils. The second line of defense is non-specific, relies on phagocytosis and is followed by inflammation. And in this context, phagocytosis is the ingestion and digestion of pathogens. So, what are the cells of the innate immune system in charge of the second line of defense? The first type of cells, which is quite relevant, are mast cells, and mast cells are in charge of releasing histamine. Second type of cells that we need to know about are natural killer cells, short NK cells. They are able to recognize and kill infected host cells. So those cells are not actually targeting the microbes, they are targeting infected host cells. Then we have basophils, eosinophils, and neutrophils, and those cells can be grouped and referred to as granulocytes based on the microscopic appearance. As you can see here in this schematic view of those granulocytes, they are full of subcellular structures that look like granules under the microscope. Those are white blood cells with granular staining pattern. They release digestive enzymes, but also histamine and heparin. Macrophages and dendritic cells are phagocytes, and actually neutrophils do belong to this group as well. Phagocytes engulf and destroy the microbes actively and release cytokines. And this is quite important to keep in mind, that phagocytes actively target the microbes, whereas NK cells recognize infected cells and removed from them from the system. But they are dealing with infected host cells and not directly with the microbes. With this, I'm closing the session number 2, and I'll see you shortly in session number 3. Thank you very much for your attention.